Hello children, I'm Ravichandra, Sri Chaitanya faculty. Here I'm there to explain biological classification. Hope you have done your board exams effectively and properly geared up for the NEET exam. Every second from now on should be properly decided and divided. No wastage of time is allowed, and I will ensure that this topic which you are listening to me now is properly described and explained so that you can remember these things without any problem. So let us start the topic. And the questions asked in biological classification approximately is from 4 to 6. It differs from one year to another. And out of all the subtopics of biological classification, Kingdom Fungi, which is one of the difficult part, has the highest weightage. And these are the questions which you will be seeing in many books. So I don't want to waste my time by talking about the questions here. Mycoplasmas contain cell wall, so they doesn't have a cell wall, so therefore that statement is wrong. And these are the examples and the characters and classification. Mode of nutrition is not the criteria for fungi. And the most common names in fungi, the common names and the scientific names and the utility is the most commonly asked question. The asexual spores of Deuteromycetes are conidiospores. And this is a very good question, and please go through it after some time. Mad cow disease in cattle, and this is caused by prions, which contain only the virus protein code, doesn't have any genetic material. Now, it's time for us to talk this topic in detail, right? So we'll start this topic, the biological classification part. We'll divide this topic, children, into 5 categories. First is introduction and different types of classification. Then we'll be discussing about Monera, Protista, fungi, and viruses. So without wasting time, we'll start with types of classification. Broadly, there are 2 types of classification, scientific and non-scientific. If you take utility of plants, useful, unuseful, poisonous, as a criteria for classification, it comes under non-scientific classification. But if you take a particular criteria for classification, it comes under scientific. There are 4 different types of scientific classifications in our textbook. The first one is Aristotle's classification. Second one is Linnaeus. Third one is Whittaker. And the fourth one is Carl Woese. Let us start with the first classification, Aristotle's classification. Criteria, nature of stem. Morphological character, nature of stem. If the stem is soft, herb. If the stem becomes woody and bushy, shrub. If the stem is woody with a trunk, tree. He also classified animals, those with red blood and those without red blood. Now, the next type of classification is Linnaeus classification. What are the criteria for Linnaeus classification? Presence or absence of cell wall. Those with cell wall are called plants. Those without cell wall are called animals. And what are the merits of Linnaeus classification? Majority of plants are grouped properly, are included in his classification, and one more merit could be scientific. It is based on science. And the demerits: prokaryotes are kept along with eukaryotes. You'll be knowing it in 2 minutes. Unicellular organisms are kept along with multicellular. Autotrophic organisms are kept along with heterotrophic organisms. So if you try to list out which organisms are kept under plant kingdom and which organisms are kept under animal kingdom, the table looks like this. Now just concentrate here. Now this is about the different classifications I talked about. Aristotle, Linnaeus, and Whittaker, 5 kingdom classification. Carl Woese separated Archaebacteria and gave a 6 kingdom classification. Now, I was talking about Linnaeus classification. It is a 2 kingdom. Criteria is based on cell wall. Merits, scientific, easy to understand. Demerits, large number of organisms doesn't fall into any category. Prokaryotes are not distinguished into eukaryotes. Unicellular organisms are kept along with multicellular, and photosynthetic organisms are kept along with non-photosynthetic. Look at Linnaeus classification. Bacteria contain peptidoglycan, therefore plant because it contains cell wall. Blue-green alga contain peptidoglycan, therefore it's a plant. Diatoms contain silica in their cell wall, therefore it is a plant. Dinoflagellate contain thick cellulosic plates, therefore it's an animal. Therefore it's a plant. Euglenoids and slime molds, they doesn't have a cell wall. He did not keep them under animals. He did not classify them because they show both plant and animal characters. Protozoans, that doesn't have a cell wall, therefore animals. Fungi, they contain chitin, which is an amino sugar called NAG, therefore it's a plant. Yeast is also a fungi, therefore a plant. Chlorella, Chlamydomonas, Spirogyra, they're all plants. These are the classification given by Linnaeus. And Linnaeus classification has many demerits. So therefore, the next classification, without wasting any time, is Whittaker's classification. In the year 1969, it is a phylogenetic classification. There are 5 kingdoms. The criteria is cell structure, cell structure means prokaryotic or eukaryotic. Body organization means does they have cells, tissues, organs or organ systems. Nutrition means are they autotrophic or heterotrophic. Reproduction and phylogeny. The merits of Whittaker's classification: prokaryotes are properly got separated and given under Kingdom Monera. Unicellular eukaryotes are given a separate kingdom called Protista. A heterotrophic organisms, fungi, are separated and given a separate kingdom called fungi. Of course, there are demerits even in Whittaker's classification. Lichens are kept. Lichens, viruses, prions and viroids did not find any place. Just now I told you, bacteria, blue-green alga, mycoplasmas and spirulina, they have primitive nucleus, therefore they are monerans. Diatoms, dinoflagellates, euglenoids, slime molds, protozoans, chlorella and chlamydomonas. Be careful, these 2 examples are important. They are unicellular and eukaryotic. That is the reason they are protistans. Fungi, they are heterotrophic, separate kingdom fungi. Yeast also is even though yeast is unicellular, it has got chitinous wall. That is the reason it is kept under fungi, it is not kept under protista. And Spirogyra is a plant, not spirulina. Spirulina is a blue-green alga, therefore it's a moneran. Porifera to mammals is animals. So these are the points with regard to Whittaker's classification. So if I just put both Linnaeus and Whittaker's classification side by side, bacteria because of cell wall plants, Whittaker because of prokaryotic nature Monera. Diatoms, dinoflagellates, chlorella, chlamydomonas, they are plants because they contain cell wall. They are unicellular and eukaryotic, therefore they are protista. Euglenoids and slime molds are not kept in any kingdom, but they are protistans according to Whittaker because they are unicellular and eukaryotic. Same is the case with protozoans. Animals because it doesn't have cell wall and protista. Spirulina, it is a blue-green alga, therefore it is monerans according to Whittaker. Spirogyra is plant. If I just elaborate this, all the subheadings here: cell structure, Monera prokaryotic, the remaining all are eukaryotic. Body organization, Monera contain only cells, Protista also contain only cells, fungi cells and loose tissue, plants contain cells, tissues and organs, animals contain cells, tissues, organs and organ systems. Coming to nutrition, Monera autotrophic and heterotrophic, same is the case with protistans. Fungi are heterotrophic, plants are autotrophic, animals are heterotrophic. Your textbook says even saprophytic. Whittaker classification gave more importance to nutrition with regard to the other criteria. Nothing wrong with reproduction, nothing special. They show asexual and sexual, phylogeny, primitive and advanced. Calvo's classification is similar to Whittaker's classification, except that Archaebacteria are separated and studied under a separate kingdom. So therefore there are 6 kingdoms or 3 domains in Calvo's classification. Now we are going to start with the first kingdom, that is Kingdom Monera. The 5 criteria given by Whittaker: first, cell structure, it is prokaryotic, primitive nucleus. Body organization, they are unicellular, no question of tissues, organs and organ systems. Nutrition, Huge amount of nutritional diversity. They are both autotrophic and heterotrophic. Complexity with nutrition is seen in this kingdom. Reproduction, mostly asexual, divided by binary fission, rarely sexual. A primitive type of DNA transfer is observed. And phylogeny, they are the most primitive creatures in the entire organisms. Now, broadly we can classify the Kingdom Monera into two units. One is Archaebacteria and other is Eubacteria. What is Archaebacteria? They are primitive type of bacteria which contain a distinct cell wall material, that is pseudomurine. And that is the reason why they can survive in the most hostile environments. The second type of bacteria, what we come across, is the Eubacteria, which are said to be true bacteria. EU stands for true. And Eubacteria are again divided into two types based on nutrition, into autotrophs and heterotrophs. Those which can synthesize their own food, utilizing atmospheric carbon dioxide, are said to be autotrophs. Need not be always photosynthetic. Mind this, children. Because chemoautotrophs, they can synthesize their own food, but there is no photosynthesis there. Heterotrophs are those organisms which depend on others, which are not photosynthetic. And under autotrophs, we come across two, photoautotrophs and chemoautotrophs. Example for photoautotrophs are cyanobacteria, which shows oxygenic photosynthesis, even chlorobium and chromatium, which come under anoxygenic photosynthetic organisms, because Von Neal classified them as anoxygenic photosynthetic organisms because H2S is the substrate which get oxidized, not water. Coming to chemoautotrophs, they oxidize inorganic chemicals. In that process, energy is released, and that energy is used to synthesize ATP. Iron bacteria oxidize iron, sulfur bacteria oxidize sulfur, hydrogen bacteria oxidize hydrogen. In this way, we can classify the chemoautotrophs into various groups, and they help in recycling of minerals. Coming to heterotrophs, they may be saprophytes, parasites, or symbionts. If they feed on dead, you call them as saprophytes. Wonderful example in your textbook is Lactobacillus, which is responsible for curdling of milk. The second example, what we have is parasite. They are feeding on living. The good example in your textbook is canker disease caused in citrus. Canker is just like a swelling, just like a cancer. All right, a tumor which is formed by Xanthomonas citri. It is an example for parasitic. Example for symbiotic is very popular, Rhizobium. It stays in the taproots of legume plants, which can do the job of nitrogen fixation, equal mutual association between legume plant and Rhizobium. Now, if we talk about each group of these in detail, I already told you, Archaebacteria, the cell wall is pseudomurine. The cell membrane also contains branched chain lipids. Histone-like proteins can be present. They are commonly called extremophiles. They may be thermoacidophiles. They may be halophiles, or they may be also methanogens. And the next group I told you is photoautotrophs. Wonderful example for photoautotrophs is cyanobacteria. They show oxygenic photosynthesis. Important points about cyanobacteria: water blooms. They may be unicellular, colonial, or filamentous. They may be terrestrial or aquatic. This point is important: oxygenic photosynthesis. They contain nitrogen fixing heterocysts. They are used as biofertilizer in rice fields. Symbiotic association in cycas. Chlorophyll A is present in chromatophores. That's a very important point in cytology which is given there. Spirulina is used as single cell protein, substitute to the normal proteins what you consume. And the next type, what we come across is other two examples of photoautotrophs, chlorobium and chromatium, which shows anoxygenic photosynthesis. Then we talked about chemoautotrophs, oxidation of inorganic compounds to synthesize ATP. Then we talked about heterotrophic organisms. Saprophyte is Lactobacillus, parasite is canker Xanthomonas, and Streptomyces is antibiotic producer. And other group which is very interesting is Mycoplasmas. They are nothing but bacteria without any cell wall. They are known to be the smallest known creatures. Mycoplasma can cause diseases both in plants and animals. And they are mostly aerobic, but they can survive even in the absence of oxygen. So they are facultative anaerobes, means sometimes anaerobes, mostly aerobic, sometimes they're anaerobic. That is a very good question which can come. And let me also tell you some of the important characters of monerans which you have to remember. And those characters are listed here under a cell structure. They can habitat almost at every place. They're simple in structure but complex in behavior because of nutritional diversity. The sole members are bacteria. Prokaryotic kingdom, the cell wall contains two types of sugars, NAG and NAM within peptidoglycan. Membrane-bound organelles are absent. Ribosomes are of 70S type. Chromatophores are present in blue-green alga which contain photosynthetic pigments, namely chlorophyll A. Nuclear membrane and nucleolus are absent. In place of it, you see nucleoid which contains circular double-stranded DNA. And plasmids are extra chromosomal DNA fragments which are used in genetic engineering. Flagella doesn't have 9+2 arrangement of microtubules. It starts from Protista. Chlorophyll A is present. Phycocyanin is present in blue-green alga. And in green sulfur bacteria, other types of chlorophyll is present. And these are the points which we already discussed. And Coccus, Bacillus, Vibrio and Spirella are the four types of bacteria based on their shape. And the most important point is Coccus is without any flagella, unlike the remaining types. It is time for us to talk about the next kingdom, which is called Protista. It is a unicellular eukaryotic kingdom. All the organisms which are unicellular with true nuclear membrane are kept in this kingdom called Protista. And what are the very unique features of Protista? I have listed here properly. And look at this. The first important unique feature is tissues, organs and organ systems are absent. Just like Monera, they contain only cells. Boundaries are not well defined because diatoms and dinoflagellates, they resemble plants. Euglenoids, they resemble both plants and animals. Slime moths, they resemble plants, animals and fungi, whereas protozoans, they are mostly animal-like protists. And they form a link with plants, animals and fungi, and mostly they are aquatic is an important point to remember. Plant protists, diatoms, dinoflagellates, plants, animal protists, euglenoids, plant, animal and fungal protists is slime moths, and animal protists are protozoans. And green protists are diatoms, dinoflagellates and euglenoids because they contain chlorophyll. Non-green protists are slime moths and protozoans. Cell wall is seen in diatoms and dinoflagellates. Cell wall in spores is seen in slime moths. Cell wall is absent in euglenoids, slime moths and protozoans. Flagella are seen in dinoflagellates, euglenoids and protozoans. Two unequal flagella is seen in dinoflagellates and euglenoids, whereas in Trypanosoma, a protozoan, there is a single flagella. Flagella is totally absent in diatoms, slime moths and the other type of protozoans except the flagellated ones. Phytoplanktons are diatoms and dinoflagellates. Primary producers, diatoms and dinoflagellates. Chlorophyll A and C, diatoms and dinoflagellates. Mixotrophic nutrition, both autotrophic and heterotrophic, euglenoids. Chlorophyll A and B, just like higher plants, is seen in euglenoids. Multinucleate plasmodium is seen in the members of slime moths. And different locomotory organs are seen in protozoans like cilia, flagella, etc. First time seen, this is an important point. 9+2 arrangement of microtubules in flagella. First time gametes are seen. First time zygote is seen. First time isogamy, anisogamy and oogamy is seen, which you don't come across in Monera. Children, I want to make this Protista look very easy. There are some side headings on which we are going to discuss all the four types of plant groups. What are the side headings? Common name, habitat, cell wall, chloroplast, nucleus, nutrition, flagella, reproduction and a special character. Let us start with the first group, that is diatoms. They are plant protists or phytoplanktons. Habitat, they are mostly marine and freshwater, an important point. Cell wall, they contain silica in their cell walls, which forms diatomaceous earth once they sink to the bottom of the sea. Chloroplasts, they are observed with A and C pigments just like brown algae. Nucleus, these have true nuclear membrane and nucleolus, hence we say them true nucleus. Nutrition, they are the first major producers of ocean, they are photosynthetic. Flagella are absent. Reproduction, both asexual and sexual. Oxospores, an outside chance for this question to come. And the special features are diatomaceous earth, passive movement of water because they doesn't have flagella, and they are pollution indicators. The next group of protistans are dinoflagellates. Common name, plant protist. Habitat, they are mostly marine. Cell wall, cellulosic walls, thick cellulosic plates. Chloroplast, they have chlorophyll A and C. Nutrition, they are photosynthetic. Nucleus is mesokaryon. They have nuclear membrane, but they doesn't have histones. That is the reason it is in between prokaryotic and eukaryotic. Flagella, two, unequal, perpendicular. When both the flagella are going to beat in water, they allow the organism to move on the water, whirling whips, very important point. Reproduction, both asexual and sexual. Special features, many types of xanthophylls giving various colors, and red tide causing one is goniolax. Goniolax is an organism which moves in huge numbers in water. They can clog the gills of fishes in that process. They can also secrete some neurotoxins like saxitoxin, which can cause the death of aquatic animals. So that's an important point. Please remember that. Euglenoids, plant animal protist. Habitat, they're standing fresh water. Cell wall is absent. Chloroplasts with A and B, very important. They are seen in higher plants also. Nutrition, in the presence of light, photosynthetic. In the absence of light, they show heterotrophic nutrition. True nucleus is seen. Flagella, one is very short, other is long, which comes out of the reservoir. Reproduction, asexual and sexual. Special features, myonemes and pellicle, astaxanthin and stigma, which allow photoreceptors, allow euglena to move towards light. Two types of nutrition, two types of locomotion, reservoir, paraflagella, arboric. Comes to the next one, that is slime moulds. They are plant, animal, fungal protists. I'll tell you why. Common name, just now I told you. Habitat, they grow on dead and decayed organic matter. Cell wall is absent around plasmodium, but cellulosic walls are present around spores. This is an important point to remember. Chloroplasts are absent. Nutrition is mostly saprophytic. The plasmodium is going to feed on dead and decayed organic matter. Nucleus, true nucleus, multinucleate condition is seen in the vegetative mass called plasmodium. Flagella are absent. Reproduction both sexual and asexual. What actually happens here? Just listen here. This small diagram can help you to understand this concept. There are two amoeba-like cells. You call them as mixed amoebae. Think that they're amoeba. Two mixed amoebae fuse. Plasmogamy occur, followed by karyogamy immediately, resulting in the formation of a diploid cell. You can call it as a diploid mixed amoeba. The diploid amoeba-like cell is going to increase in its mass, undergo many free nuclear divisions, and develop into a multinucleate vegetative mass called plasmodium. This plasmodium is diploid in nature because the nucleus is diploid. It is going to secrete slime when it moves on the ground, on the dead and decayed organic matter. What happens to this plasmodium when the conditions are not favorable? This plasmodium produces aerial sporangium, means it extends out and develops into a fruiting body called sporangium. All the diploid nuclei migrate into sporangium. They undergo reduction division and give rise to spores. This is asexual reproduction. Spores are with cellulosic walls, a plant character. Sporangium is seen, a fungal character. Plasmodium is without cell wall, animal character. That is the reason we say plant, animal, fungal protists. Now let us come back to spores. What happens to spores? Spores get liberated, they are windborne. Once they reach the soil, 50% of the spores, the wall break and develop into mixed amoeba of plus strain. The remaining 50% of the cells will develop into mixed amoeba of minus strain. This is the life cycle of slime molds, which is nicely explained. Hope that you understood it. Last group is protozoans. which are animal protists based on the locomotory organ. Amoeboid protozoans, they doesn't have cilia or flagella, they contain pseudopodia which allow it to move. Second type is flagellated protozoans. Example for that is Trypanosoma, Leishmania. They contain a single flagellum. The third type is a ciliated protist. And the example for ciliated protist is Paramecium. And the most important part about Paramecium, which was asked previously, it has got two nucleus, one micronucleus and one macronucleus. And it contains many cilia, which are going to help in a synchronous movement, allowing the water and food to get into the gullet. The last type of protozoan is a sporozoan, which produces a spore-like stage called sporozoid. And this sporozoans, wonderful example for that is a malarial parasite, that is Plasmodium. That completes the protozoans, which is the animal protists. Now we'll be talking about the most important kingdom called fungi. If there is any area, troublesome area in the entire biological classification, this could be one reason. So listen carefully and see that you don't miss any point. Same way, we'll take the 5 criteria given by Whittaker. First is cell structure. They are eukaryotic. Cellular organization. They contain cells and loose tissues. Nutrition. They are only heterotrophic. No question of autotrophic nutrition here. Reproduction. They show asexual spores like sporangiospores, zoospores. And at the same time, they show sexual reproduction, which could be a primitive type, like in Ascomycetes and Basidiomycetes, very properly seen as you see in Phycomycetes, or totally absent as you see in Deuteromycetes. Let us elaborate these 5 criteria given by Whittaker a little bit more. First, I come back to cell structure. Cell wall is made up of chitin, which contain amino sugar, NAG, and also contain polysaccharides. Plasma membrane, just like higher plants, contain lipids and proteins. Cytoplasm is semi-fluid matrix. Ribosomes, there are 80S in mitochondria, and even 70S can also be seen. All right, then in mitochondria, 80S in cytoplasm, 70S in mitochondria, correction. Reservoir, it can have glycogen or oil. So that's about the cell structure. Coming to body organization, already I told you, body organization, they are unicellular. And if you look at, they are aseptate and multinucleate in Phycomycetes. Nice question. When the septa are absent, there are many nuclei under a common compound. This condition is said to be aseptate and multinucleate. That is seen in the members of Phycomycetes. Cenocytic is the right word to be used there. Septate, uninucleate and binucleate. In Ascomycetes and Basidiomycetes, initially the hyphae are uninucleate, but after plasmogamy and before karyogamy, they may be binucleate as well. So both uninucleate and binucleate condition is seen, but mind you, both the conditions are haploid except after karyogamy. And in Deuteromycetes, there is no question of binucleate mycelium. It is just like uninucleate mycelium only. And nothing for me to talk about nutrition. Already I told you it's a heterotrophic nutrition. Now we'll talk about few important points about fungi. They prefer warm and humid regions. Linnaeus classification, they are kept under plants because they contain cell wall chitin, which contain N-acetyl glucosamine as one of its monomer. Whittaker's classification, they are given a separate kingdom called fungi. So a lot of good treatment is given to fungi by Whittaker, unlike Linnaeus. Vegetative body, long thread-like hyphae, which forms the mycelium. Without wasting any time, now it is asexual spores, already I told you, sporangiospores and zoospores. In Ascomycetes, conidiospores, in Deuteromycetes, also conidiospores. Sexual reproduction involves plasmogamy, karyogamy, and meiosis. Anyway, I'll come back to this topic a little later. We'll start with the first class of fungi, that is Phycomycetes. What are the side headings on which we're going to discuss? First is habitat. They are aquatic. They can also be on decayed wood, damp places, and they can also be obligate parasites. Mycelium. They doesn't have any septa, aseptate and cenocytic. Vegetative reproduction is by fragmentation. Asexual reproduction, you know what? Some of the hyphae, they are negatively geotrophic, and they are going to produce a terminal sporangium. And all the nuclei migrate into the terminal sporangium and transform and produce a wall and transform into spores. If they are non-motile, you call them as sporangiospores. If they are motile with 2 flagella, you call them as zoospores. And the point to remember is both these asexual spores are endogenous. They are produced inside the sporangium. Remember this very important point. Sexual reproduction, I told you, it is the true sexual reproduction. Very important. What is the meaning of true sexual reproduction? It means that these hyphae are going to produce gametangia. Gametangia are going to produce gametes. Gametes fuse to form zygote. Actually, gametes undergo fusion. Plasmogamy and karyogamy occur immediately, resulting in the formation of many diploid nuclei. That structure with many diploid nuclei is called zygospore. The nuclei undergo reduction division and give rise to the spores. And those spores are called sexual spores. You call them as germ spores. And these germ spores get liberated. Shall I tell you? Sporangiospores and germ spores, the production is more or less the same inside the sporangium. The only thing is that sporangiospores are asexual spores, whereas germ spores are sexual spores. That is the big difference. And the examples, Rhizopus is a bread mold, Muca, similar to Rhizopus except that it doesn't have rhizoids. Albigo, it causes white rust disease in mustard. Phytoptera, Irish famine. All right, people started thinking about the diseases in plants for the first time when the famine struck Ireland. All right, and that is the Phytoptera causing huge losses in potato farmers. Then we talk about the next class, children, without wasting any time, Ascomycetes. Again, common name sac fungi, habitat coprophilous, parasitic, mycelium. They may be unicellular, multicellular, septate, monokaryotic or later dikaryotic. Vegetative reproduction is fragmentation. Asexual reproduction is by conidiospores. This point is important. It is exogenous in origin. Coming to sexual reproduction, let me explain this in the form of a diagram. Concentrate here. Your textbook did not put all the things in the right perspective. So you can understand it better if you follow what I'm saying. Listen, the hyphae of plus strain, this is my left hand is the hyphae of plus strain, my right hand is hyphae of minus strain. The hyphae of plus strain is going to produce a sex organ called antheridium. The hyphae of minus strain produce a sex organ called ascagonium. Now you know what happens? A small tube extends from antheridium to ascagonium. Now the nuclei migrate. Listen carefully. Nuclei migrate, but the nuclei will not fuse with the nuclei of ascagonium. They only migrate. It means that plasmogamy occurs between antheridium and ascagonium, but not karyogamy, resulting in the formation of a dikaryotic nucleus or a dikaryotic cell. This undergo many mitotic divisions and develop into a cup-like fruiting body, and that cup-like fruiting body, you call it by one name, that is said to be mushroom or ascocarp. What will happen to this ascocarp? The ascocarp definitely contain all binucleate cells. One of the binucleate cell is sac-like and it is called sac fungi. And the two nuclei inside that first time fuse, resulting in the formation of a diploid nucleus, and that is followed by reduction division immediately, resulting in the formation of 4 sexual spores called ascospores. And later they become 8. The ascospores are endogenous inside the ascus. The ascospores get liberated. 50% of ascospores develop into hyphae of plus strain. The remaining 50% will develop into hyphae of minus strain. Same, the hyphae of plus strain produce antheridia, minus strain produce ascagonia. Plasmogamy occur but not karyogamy. You see the formation of a cup-like ascocarp, then ascus, karyogamy occurring here, reduction division occurring in ascus, producing spores. Examples are important. Saccharomyces, baking and brewing industry because it secretes zymase. Penicillium, antibiotic producer. Neurospora is an experimental fungi. Aspergillus in microbes chapter is given, it produces citric acid. Claviceps, it causes ergot disease in rye. Morcella and tuber are examples of costly mushrooms. The common names are given, morels and truffles in a textbook. Gibberella in plant hormones is given. Foolish seedling disease. Monascus produce statins. That's about Ascomycetes. Now we talk about Basidiomycetes. Shall I tell you? Once you understand Ascomycetes, Basidiomycetes looks very simple. The only thing is that you don't see the sex organs. That's all. The remaining is same, exactly same. You'll discover that in a minute. Common name, club fungi. Habitat, soil, logs, tree stumps. Mycelium, it is multicellular, septate, monokaryotic and later dikaryotic. Vegetative reproduction is by fragmentation. Asexual reproduction is absent. Sexual reproduction is a primitive type. Just now I told you, listen here. The hyphae are plus and minus strain. They fuse, no sex organs. Plasmogamy occur again, but no karyogamy, resulting in the formation of a dikaryotic cell. It undergo many mitotic divisions and develop into a mycelium. This mycelium produce aerial fruiting bodies, the one what you eat. That is mushroom. It contains a stalk and a cap. On the cap, on the lower side, you see the presence of gills. If you take a section of a gill, you're going to see a club-like structure called basidium. Hence, it is called club fungi. The two nuclei fuse, reduction division, basidiospores. Unlike ascospores, basidiospores are produced exogenously, you could see there, and they get liberated. So doesn't it look like Ascomycetes, except that there are no sex organs here. The vegetative hyphae fuse, hence called somatogenous copulation. Amanita is a poisonous mushroom. Eustylago, smut causing. Puccinia, rust causing. Lycoperidon is a puffball. Polyporus is a bracket fungi. Clamp connections and dolypores are there in dikaryotic secondary mycelium. Nothing for me to talk about Deuteromycetes. It's fungi imperfecti. It grows in dead and decayed organic matter. Mycelium is septate and branched. Vegetative reproduction is by fragmentation. Asexual reproduction, just like Ascomycetes, is with the help of coniidiospores. True sexual reproduction is absent. When any sexual form of this fungi is discovered, then they are moved to a class rightly belong to. I'm going to give an example here. Fusarium moniliformis. This is an example of a Deuteromycetes member. Once it started showing some sexual stages, sex organs, etc., immediately it is shifted to Ascomycetes with a separate name called Gibberella fusicori. And the common examples, Alternaria, early blight in brinjal, Colitotrichum, red rot disease in sugarcane, Trichoderma. It produces cyclosporin A, which is an immunosuppressive agent or a biocontrol agent. Now we're going to talk about the last group under the biological classification, viruses. The reason why we're discussing viruses is Whittaker did not keep these viruses in any of the kingdoms because it doesn't show any cells. And what is the definition of viruses? They're ultra-microscopic obligate parasites. Why you say ultra-microscopic? Because they're very, very small. Why you call them as obligate parasites? The reason why they're called obligate parasites is because they require a living host for their multiplication. So they doesn't depend on host for trivial water or food. They depend on the host for their metabolism, or for their reproduction. They doesn't have any metabolism, reproduction. So if I'm a virus, I have a protein coat and I have a genetic material. So I require ribosomes in order to produce protein. I don't have any cells, no question of ribosomes in me. So therefore I'm in search of a cell with ribosome. So it could be a plant, it could be an animal, it could be a blue-green alga, it could be a fungi. So whichever cell contains ribosome, I go into them and I use their ribosomes, I use their enzymes for my multiplication. That is the reason viruses are example for obligate compulsory parasites. So without any living host, they cannot survive, they cannot multiply, is an important point to remember. What are the non-living characters of viruses? They doesn't have any cells. That is the reason they don't have any metabolism. If you're suffering with any viral disease, no antibiotic can cure you. So if you're suffering with cold, if you go to a doctor, it takes 7 days. If it doesn't go to a doctor, it takes 1 week. It means that whether you go or doesn't go, it doesn't make any difference, because viruses doesn't have any metabolism, doesn't have any cellular mass which can get affected by an antibiotic. They are not influenced by antibiotics. They cannot be cultured. Naturally, they cannot be cultured. Any living things can be cultured. Viruses cannot be cultured. Then if you want to culture a virus, you have to culture a cell which contains a virus. So if you culture a cell which contains virus, you can even culture viruses. But viruses on separation cannot be cultured. And absence of intrinsic motility is also a non-living character. If you look at living characters, they contain the genetic material. It could be either DNA or RNA. Not both. If you look at some viruses, plant viruses, if you look, they mostly contain RNA. Of course, cauliflower mosaic virus and dahlia mosaic virus contain DNA. If you look at animal viruses, the meaning of animal viruses, those viruses which resemble animals? No. Those viruses which infect animals. Animal viruses generally contain DNA as a genetic material. Having said that, you have got some animal viruses which you study in your zoology. All right, HIV, which contain 2 RNA molecules, coronavirus, which you know about it few years back. We had the MNAs of this virus which contain RNA as a genetic material. But having said that, majority of animal viruses contain DNA as a genetic material. Bacteriophage, they also generally contain DNA as a genetic material. Having said that, QB bacteriophage and molecular basis you studied, they have a RNA, single-stranded RNA as a genetic material. It is also very interesting to know that viruses are the only group which has got double-stranded RNA and a single-stranded DNA. Interesting. Single-stranded DNA is seen in 5x174 bacteriophage. Double-stranded RNA is seen in reovirus, wound tumor virus, rice dwarf virus, maize dwarf virus, blue tongue virus. These are all examples which has got so diversification with respect to number of strands, and either DNA or RNA is very much seen in viruses as an important point. And what does a virus contain? It is a nucleoprotein. It contains a protein coat called capsid, and it contains a genetic material. I already discussed about genetic material. Coming back to protein coat, it is called capsid and it's made up of many subunits called capsomeres. In TMV, tobacco mosaic virus, all the capsomeres are arranged in a helical manner, helical symmetric. But if you look at bacteriophage, the head part of it is hexagonal, in which the capsomeres are arranged in cubic symmetry, and the tail part of it has a helical symmetry. So it is nothing but 2 types of symmetries are observed in a bacteriophage. And the bacteriophage also contain a single tail plate and 6 tail fibers. And the tail fibers contain an enzyme called lysozyme, which can dissolve the peptidoglycan of bacteria. And the tail fibers are also called prongs. It is given in the diagram in microbes chapter. That's about the structure. Now we talk about the scientists. Very important. Ivanovsky, when he was studying about tobacco mosaic virus, they were able to pass through bacterial filters used to separate bacteria. So the reason why they're able to pass through bacterial filters, because they're smaller than bacteria. Hence Ivanovsky called it as filterable agents. Bijonic called it as contagium vivum fluidum, because they're contagious living fluid. He took a diseased leaf and he rubbed on a healthy leaf. The healthy leaf also suffered with it. And it's very common observation that viral diseases are mostly contagious. If a person in your house suffers with cold, there's a probability of the entire family suffering with it. So same is the case with TMV, it's highly contagious. So contagium vivum fluidum, this word was used by Bijonic. Stanley for the first time crystallized TMV. The meaning of crystallized is separating it and studying it. So understand the meaning of crystallization there. And bacteriophage lambda has a tadpole shaped, it contains a head hexagonal, and it contains double-stranded DNA inside the head. It contains a collar, no specific job, just like the collar in my shirt. Then it has got a tail, and it has got 1 tail plate, and you have got 6 tail fibers. And what are the plant diseases caused by viruses? Mosaic, vein clearing, leaf roll, leaf curl, and chlorosis. What are the animal diseases caused by virus? Smallpox, chickenpox, polio, measles, flu, hepatitis, AIDS, corona, mumps, herpes. They're all the diseases which are caused by viruses in animals. Viruses can be used as vector in genetic engineering, and baculoviruses are biocontrol agent in integrated pest management. So they target a specific arthropods and insects, not all. Prions, they are those virus-like particles. Remember, they are not viruses. Virus-like particles, almost the same size of viruses, but they doesn't have the genetic material. They have only protein coat, which is highly infectious. Mad cow disease or BSA in cattle and CJ disease in humans are the most common diseases caused by prions. Viroids, they're smaller than viruses. Scientist's name is Diner. They have a single-stranded RNA in them. And the potato spindle tuber and citrus hexocortosis disease are the 2 diseases which are caused by viroids. Then coming to lichens. The reason why Whittaker did not put lichens in any kingdom, even though they are cellular, is the fact that it has a colony of alga and fungi staying together. He doesn't know whether I should keep it in plant kingdom or in fungi. So he kept it separate, and that is lichens. So it is cellular but didn't find any place. Colony of algal and fungal components. Algal members can be Chlorophyceae or Cyanobacteria. Fungal members can be Ascomycetes or Basidiomycetes. Do they fix atmospheric nitrogen? You can't stop them from fixing atmospheric nitrogen because Cyanobacteria are present here. They are pollution indicators of sulfur dioxide. They are the pioneer community of an ecological succession operating on rocks. And what about diatoms and dinoflagellates? They are the first community of phytoplanktons on the ecological succession operating in water. So that is called pioneer community. And examples, Cladonia and Roxella are the two examples outside chance for this Roxella, litmus producing moss. That completes the last part of biological classification. Hope that you understood this entire topic of biological classification properly. If this is little done in a haste, then I want you to go back and listen one more time. All right, go through the tables and the pages which I've kept here. That will be extremely handy. Answer an assignment based on it. Then you'll be extremely good. All the very best, and we'll meet you again. Thank you.